Hi everyone, welcome back to my educational channel on biology. In this video, we'll discuss another technique to estimate the population size of organisms, other than the quadrant sampling technique. Now this technique is called the Capture, Mark, Release, Recapture technique. In short, it's CMRR technique, right? So it's used to estimate the population size of organisms, especially animals that can move freely in the area of study. Examples of such animals are birds, for example, like this kingfisher here, snails, rats, even fishes like sharks, you see, invertebrates such as insects like butterflies, and also millipedes and woodlice. So all these can move freely in the area of study, and they can mix with the other individuals of the same population. Right, so clear explanations will be given, and at the end of this video, this short video, we have excellent questions which help you to train you to apply the formula and calculate the population size of organisms. So let's begin. The learning standards are as follows. Chapter 9, Ecosystem. 9.2, Population Ecology. We should be able to analyze data to estimate the population size of organisms using, number one, the quadrant sampling technique. This has been discussed in the previous video. So in this video, we'll discuss the Capture, Mark, Release, Recapture technique. Right, so here's our concept map. We are going to discuss the Capture, Mark, Release, Recapture technique, which is used to estimate the population size of animals that can move freely in their habitat, such as rats, even crabs in the mangrove swamps, and so forth. Okay, so the CMRR technique, or Capture, Mark, Release, Recapture technique. Now, what is this technique called the Capture, Mark, Release, Recapture technique? So the Capture, Mark, Release, Recapture technique, or CMRR technique, is used to estimate the population size of organisms or animals such as snails, rats, birds, foxes, fish, for example, sharks, and invertebrates even, like insects, such as butterflies, millipedes, and woodlice. Now, millipedes and woodlice are invertebrates that live in the soil. These animals can move freely in the area of study. So they must be able to move freely so that the marked animals can mix with the unmarked ones. All right, before the second capture. So let's find out the steps of the CMRR technique. Firstly, animals of the species under study, all the animals of the same species. Now, they are captured randomly. So you can capture a number of these animals in the area of study, which can be the natural habitat or in that particular environment. Okay, so a number of the animals of the species under study are captured randomly. Randomly means that when they're captured, we don't plan to capture those with particular characteristic, like of a certain size or of a certain color and so forth. Whichever animal of that species that you see, you capture. All right. So in Malay, we say secara rawat. So you capture a number of these animals in the area of study. Now, the number of animals captured in this first capture, P, is then recorded. Okay, so we assign the letter P for the number of animals in the first capture. Let's say I'm trying to estimate the population size of snails in a field or in a vegetable plot. So let's say in a field. So I capture 10 snails. It's up to you how many snails you want to capture in the first capture. Okay. So every animal captured is marked by using non-toxic nail polish, paint, or Indian ink. Now for rats, maybe they put a tag on the neck or something like that. Okay, or for fish. So for snails, we can mark them with non-toxic nail polish, paint, or Indian ink, right? So the mark, what are the characteristics of the mark used? So the substance used to mark the animal must be non-toxic. That means not poisonous, okay, as shown here, right? So it doesn't harm the individual, and also it must be able to stay and withstand the effect of rain, so it doesn't dissolve in the rain water, okay? Also, the third characteristic is that the mark must not make the individual more obvious to predators, in case the predators will choose to eat the marked animals or individuals. Okay, so these are some characteristics of the mark or the substance used to mark the snail. So all the animals in the first capture are marked, right? Then the marked animals are then released back into the same area of study so that they can mix with their friends, okay, with the other individuals of the same species. Then after a certain period of time, a second capture is done randomly in the same area of study. So this can be a few days to a week, depends on the organism. For snails, because they move very slowly, so we need to give them enough time, okay, to move around and mix with the other individuals in the same population, right? Now, after a few days to a week, a second capture is done randomly in the same area of study. Now, this second capture is your sample. You're trying to find out a ratio or proportion of something, okay? So the total number of animals captured in the second capture Q is recorded. Let's say in the second capture, I decide to capture 20 snails. Again, this is done randomly. I don't only choose snails that have the mark, all right? Whichever snail I see, I just capture, right? So also record the number of marked animals in the second capture, R. So let's say I captured 20 snails. Then I have to observe the 20 snails. Which one has the mark on it? Let's say out of 20 snails, 5 snails were marked with a mark. Could be nail polish or paint, right? So estimate the population size of the species, of this species of organism, like snails, by using the given formula below. So this is our formula that we have to memorize. Population size equals to P, that means the number of animals in the first capture, the total number of animals in the first capture that were all marked, times Q, the total number of animals in the second capture, and then divide by R, which is the number of marked animals in the second capture. Okay, so if I use my example, it will be 10, P is 10, times 20, so Q is 20, divided by 5, because R is 5. Okay, so my answer is 40 snails. Right, so this is the formula for the capture mark release recapture technique, CMRR technique. Population size equals to P times Q over R. Or you can use other letters like X times Y over Z. Okay, so here P represents the number of animals in the first capture, Q is the number of animals in the second capture, the total number of animals in the second capture, and R is the number of marked animals in the second capture. So animals involved can be snails, rats, birds. And if you are wondering what is woodlice is a tiny invertebrate, not an insect. It has quite a number of legs. It lives under the rocks or under the pieces of wood, and it is the decomposing material in the ground. Okay, so the size is about 1 cm or so, depending on the species. So we're going to find out how did we derive this equation, so that's easier for you to remember the equation for calculations. Now, how did we obtain the CMRR technique formula? As shown here, population equals to P times Q over R, or you can use other letters like X times Y over Z, okay, where P represents the number of animals in the first capture, Q is the total number of animals in the second capture, and R is the number of marked animals in the second capture. So it's good for you to know how this equation is derived, so that in case you forget this formula, you can still formulate or form a formula yourself from which you can calculate the population size easily. Okay, so here's the explanation. In the first capture, we captured P animals and marked all of them, right, and then released them back into the area of study. That means in the total area of study, out of the total population, P animals were marked. Okay, P animals were marked. Then, after a few days, after the animals that are marked have mixed with the animals that are not marked, then you carry out the second capture. Your second capture is actually a sample, all right? and you want to find out this proportion, Q over R. You want to find out this proportion, what is the total number of animals captured per the marked animals in the second capture. Okay, so this proportion Q divided by R will be equivalent to population divided by P, meaning to say out of Q animals, R were marked. Okay, out of Q number of animals, R were marked. This fraction is similar and should mirror this fraction here. Out of the total population, P were marked. All right, so population per P equals to Q per R. Then you bring this P up to this side on the right hand side, and you get population equals to P times Q over R. Now here are three assumptions for the CMRR technique, which we make when we calculate the population size. Number one, after the first capture, the marked individuals have sufficient time to mix randomly with the whole population. So for example, if you mark the snails and then release them into the habitat, cannot just give them 1 day for them to mix randomly with the whole population. You may have to give them a few days, right? Depending on how fast they move. So you can maybe give them a few days for them to move around and mix randomly with the whole population, mix uniformly with the whole population, so that the results of your estimation of population size is more accurate. Number two, there is no change in the population size during the period of study. This means that the population size remains constant and that there are no additions to the population through births where new organisms or individuals are produced, or immigration, meaning that there are new individuals that move into the area of study from other parts. Okay, and no losses through deaths or emigration during the period of study. So in emigration, some of the individuals may move out of the habitat or the area of study. So we assume that there are no additions or losses to the population, and that the individuals in the population all stay within the area of study for the period of time that you carry out the experiment. Okay, so the second assumption is that population size remains constant throughout the period of study. Now, the marking process, this is the third point. The marking process should not affect the survival rate of the marked individuals. So we assume that the mark doesn't harm the individual and does not make them more obvious or vulnerable to the predators. All right? That means that the mark is not toxic to them. So now let's test ourselves. These are some commonly asked questions, similar to SPM questions. A group of students carried out an ecological study to estimate the population of snails in the school field. They caught 40 snails, then marked all of them and released them back into the field. After 5 days, they caught 30 snails and found that 5 were marked. What is the estimated population of snails in the field? Right, this is a structured question. So make sure that you show your working. All right, and you must also give units for this answer. Right, so try this question and also try B, state 2 assumptions that are made in this technique. Right, what is your answer for the estimated population? The answer is 40 times 30 over 5 equals to 240 snails. All right, so the population of the snails equals to the number of snails in the first capture, which is 40 snails, and then multiplied by the number of snails in the second capture, the total number of snails in the second capture, which is 30 snails, and 5 were marked, so divided by 5. Okay, so the answer is 240 snails. So these are the two workings. If you use the formula straight away, then it will be x times y over z, or p times q over r, right? Where, let's say I use x. So x is the number of snails in the first capture that were all marked, that's x, and that is 40 snails, right? And then y is the total number of snails in the second capture, which is 30 snails, right? and then 5 were marked. So 5 is this for Z. Okay, 5 Z is equals to 5. So 40 times 30 over 5, right? And the answer is 240. Or if you forgot the formula, then just remember this. Total number of snails is X. Let's say over all the marked snails in the habitat is 40, equals to this proportion. In the second capture, out of 30 snails, 5 were marked. Okay, so this equals to this. And so X equals to, you bring this over to this side, okay, to the right hand side. So you get 40 times 30 over 5 equals to 240. So even if you forgot your equation, you can still use the ratio method or proportion method to get your answer. What are two assumptions that you made in this technique? Now take note here that I've shown the working for A and also the units, which are snails or individuals. Okay, state two assumptions that are made in this technique. Now, after the first capture, the marked snails have sufficient, marked individuals or snails have sufficient time to mix randomly and uniformly with the whole population. Okay, randomly, put randomly. And number two, there's no change in the population size during the period of study. The population size remains constant. No new births or deaths or migration that affect the population size. So 1 mark for each assumption. Question 2, an objective question. An ecological project was carried out to estimate the population size of fiddler crabs in a mangrove swamp. Number of fiddler crabs captured. First capture, 40, okay, that were all marked. Second capture, 15 were marked and 35 unmarked. So what is the estimated population size of the fiddler crabs in the mangrove swamp? Okay, try this question yourself first, and we'll discuss the answer in a short while. So do be careful about the numbers that you need to use, okay, in your formula. So what is your answer? Did you get C? 133 crabs. Okay, let's see how what the working is like. Total number of crabs in the second capture, be careful, is 15 plus 35, which is equals to 50. Okay, so the total number of crabs is not 35. It's these two numbers added together, right? Now, the population of crabs in the mangrove swamp, the fiddler crabs, equals to X times Y per Z or P times Q per R, whichever one. But you must know that X stands for the number of fiddler crabs in the first capture, which is all marked. So that's 40. Y is the total number of crabs in the second capture, which is 50, not 35, 50. And then the number of marked crabs in the second capture is 50, right? So the answer is 133, right? So right, that's all for this lesson. I hope that you have you were able to understand and apply your formula to all these questions and get the correct answer, right? So please do share, like, and subscribe to my channel and comment below. So till we meet again, goodbye and stay calm and study.